Hello and good afternoon, everybody. All of you joining us here at Chalmers, and those of you joining us at YouTube. Most welcome, everybody. And a special welcome to Lena Träskov-Turell, the former chair of Chalmers, professor, and to whom this seminar is donated. As you have seen from the invitation, we will have the pleasure this afternoon to listen to a wide range of researchers from different areas. And we will also have a panel discussions about the implication that scientific results can have on society and on our lives. I would also like to encourage you all to participate in this seminar and to be active during the afternoon. And you can do that through a tool that is called the Menti, which you probably have used before. And you can see on the screen here, you can see Menti. You take your cell phone, you go to the search field and you write menti.com. And then you use the code, which is 40630755. So please do that. And then please answer the following question. Do you think that new technologies like AI, biotech, or other areas will have only a positive effect on our lives? Yes, no, or partly? And when you thought about that and you have decided, please push submit. And all the time during this seminar, you can use Menti, you can use the same code to send in comments or reactions or questions to the speakers and to the panel discussions. I hope you will do so. Before we look at the results from the first question, I would like to introduce to you now the president and the CEO of Chalmers, Stefan Bengtsson. Please give him an applause.
Thank you very much, dear everyone, dear Lena, in particular, of course, today. It's also my pleasure to welcome you to this seminar on AI, biotech, and genetics. The seminar is in honor of Professor Lena Träskov-Turell and is a gift from Chalmers when she stepped down from her position as chair of the board of directors of Chalmers. The topic of the seminar has been developed in dialogue with you, Lena. And today we will listen to outstanding scholars giving different perspectives on the theme of the seminar, and we will also have this panel discussion. So for all of you, some words about Lena. She received her PhD in physics from the University of Gothenburg, and she then worked as a senior lecturer and associate professor of physics at Chalmers. And in fact, for me, at the time, as a young student, both at University of Gothenburg in physics and then at Chalmers in engineering physics, I do remember you, Lena, as one of the teachers many, many years ago. She contributed to the construction of an advanced laser lab at Chalmers in the mid-70s, and her material physics research later focused on polymers in lithium batteries. In the late 1980s, Lena was appointed professor of solid-state physics at Uppsala University, and shortly thereafter as professor of material physics at Chalmers. She was vice president of Chalmers for 4 years in the mid-90s and was appointed head of research at the European Commission's Joint Research Center in Brussels in 1998. From 2001 to 2008, Lena was CEO of the Royal Academy of Engineering Sciences and then became the EVA chair of the board. Her deep interest in technology issues, especially technology issues in society and affecting society, where technology solves problems, have characterized her agenda through her career. Lena is or has been a board member of many other companies such as Ericsson, Investor, Mycronic, Gambro, SKF, and Saab. Lena was involved in both Chalmers and Chalmers Foundation boards for most of the 2010s and was chairman of the Chalmers board between 2013 and 2020. She has contributed with a unique combination of solid academic understanding and extensive experience from board work in both globally active listed companies and smaller companies. And now, among Lena's strong leadership qualities is her very sharp eye that is given to long-term development and to renewal of Chalmers from her work in the boards. The ability to take advantage of different perspectives in the board, create a consensus on the way forward, keep pace and achieve results even on very difficult issues. Lena, with your energy and humor, you created an atmosphere, an open atmosphere around the board table where innovative ideas were welcomed. For me personally, as being president of Chalmers, and Lena has provided continuous support during the years we worked together, encouragement, always to the point, suggestions to various questions I faced, and the ability to lift, and the ability for me to lift and discuss novel ideas with you. So Lena, thank you for all your different contributions to Chalmers. It's a pleasure to having you here for this seminar this evening, and now let the seminar begin. Thank you very much.
Thank you so much. Now let's see the results of your voting. So, do you think that new technologies like AI and biotech will affect your lives only in a positive way? No. Yes. No is 28 votes. Yes is 11 and partly 9. Well, there are questions to be asked, I suppose. That's exciting for us this afternoon to discuss more about the implications. Now I would like to introduce to you the first speaker, and that is Ross King, who is professor here at Chalmers with research focusing on machine intelligence. And he will talk about DNI supremacy and the potential of DNI-based computation. Most welcome, Ross.
Thank you. Okay, I'm going to talk about two subjects, DNA computing and the automation of science. But before I do that, I would like to say a little bit about science and technology and society, and that the world is faced with enormous problems. We have climate change, we have billions of people living in poverty, we have a pandemic, we have horrible diseases, cancer, malaria, etc. And I think if all the world got together and worked together, if our existing science and technology, that would be sufficient to solve these problems. But my reading of history suggests that's unlikely. And the only way forward then is to have better science and technology, and that's what I'm going to describe a couple of areas which I've been working on. So I'm going to talk about DNA computing and then automating science. So the idea of DNA computing is to use DNA as the substrate for computation rather than silicon. And DNA is essentially all about information. It's the information how to make other living organisms. And this was realized by the founders of molecular biology, but it's been a bit lost over the years. And DNA can store information, it can also copy information. That's how living organisms replicate, they copy DNA. And molecules are the smallest type of matter we can easily control. We can't really control subatomic particles very easily to compute. So that makes DNA an ideal substrate for computation. DNA can encode information. Instead of ones and zeros, like in a computer, we have 4 bases, which convey information, and they can be ordered in different ways to convey and encode symbols. So back of the envelope computation estimates of what you could do on the desktop compared to existing computation. So you could probably build on a desktop a DNA computer which is 10 times faster than the biggest existing computer. And if you want to see one of these big computers at the moment, they take the football-sized areas of space and power supplies, more than... My colleagues in Edinburgh, they're building a new supercomputer there, and it has a bigger power supply than Edinburgh Airport. It's crazy how much power these big computers take. So, and it could execute more operations per second than any existing computer, and the storage is far higher than any existing computer, potentially. So my colleagues and I are working on what we call DNA ascendancy. So that's building a computer that can compute something that no silicon computer could do, or even a quantum computer. And we believe that we have a plausible path how to do that. We call that DNA ascendancy. And the particular type of problem that we intend to do this on is it's technically called NP-complete problems. And it's a particular class of problems which we know that silicon computers are not very good at, and quantum computers are no better, really. So these problems, these NP-complete problems, are, I think, the most important in science and industry. They're, for example, scheduling problems. The traveling salesman is a famous example of this type of problem. And it's generally believed by physicists and computer scientists that the only way to guarantee solving these problems is through brute force search. And because DNA is very small, you can make literally billions upon billions of them to fit in the space here. You could get, I don't know, countless billions in there of computers, all acting independently, doing brute force search. And all the computation can be local, and the answer is easy to recognize once you see it. So that's our dream, to build this supercomputer to solve NP-complete problems and have DNA ascendancy faster than any other type of computer. The other area I'm going to talk about is automating science. So already AI systems have superhuman scientific powers. An AI can flawlessly remember vast numbers of facts. AI system can execute flawless logical reasoning. It can execute near optimal probabilistic reasoning. The AI systems learn more rationally than humans on small amounts of data. They can learn from vast amounts of data which no human being could ever encompass. They can extract information from millions of scientific papers. They can't read them like a human being can do. They don't get as much information out, but they can actually go through them all. No human would ever have the time to do that. So AI systems have superhuman powers which complement that of human scientists. And the traditional name for applying AI to science is called scientific discovery. And it's a very nice application area for AI. So scientific problems are quite abstract, like the games of chess and Go, which AI systems have triumphed over human beings. But it involves this very important aspect of the real world, so therefore it's much more important to do science than games. And scientific problems are restricted in scope. So that means that if you have an AI system working on a scientific problem, it doesn't need to know about other things, just that particular problem. So to quote Lewis Carroll, there's cabbages and kings and other things. You don't need to know about these. Just need to know about that scientific problem. And that simplifies it for the AI system. Another advantage is that nature is honest. By this I mean that if an AI system is doing a scientific experiment, or a human is doing a scientific experiment, we're fairly confident that the real world is not trying to mislead us about how the world works. It's giving us an honest answer to the experiment. We may misunderstand the meaning of that experimental result, but we're pretty confident it's an honest result. And that's quite different from having an AI system, say, work in the stock market or in a military application where many of the agents are dishonest. I'd also argue strongly that nature is a worthy object of our study. So many of the smartest people in the world are working on AI, but what they're doing is not solving these big problems of climate change or disease. What they're doing is making more addictive advertising for big tech companies, and that is some sort of tragedy, I think. It's bad for the world. I think it's bad for them. I wouldn't want to look back on my life at the end and just made better advertising. And the generation of scientific knowledge is a public good, I would argue. as long as that knowledge is in the public domain. So my contribution of AI to science is this concept of a robot scientist. This is an AI robotic system which can, in a sense, do autonomous scientific research. It knows something about an area of science and background knowledge that's represented in logic and probability theory. It can autonomously form novel hypotheses about that area of science. It can then autonomously decide on efficient experiments to test these hypotheses, efficient in terms of time and money. It can then program a laboratory robot to execute these experiments. It can then analyze the results of the experiments and change the probabilities on its hypotheses based on these results. And it can cycle round until it runs out of some resource or there's some theory which is consistent with the background knowledge and all the experimental results. So that's the basic idea. The first robot scientist was called Adam, and it was, we argue, the first machine to autonomously discover novel scientific knowledge. The second robot scientist, Eve, was much better designed, much more elegant, and it's in a few hundred meters that direction. We brought it to Chalmers, and it's currently working on COVID research, trying to find a small molecule to treat COVID if you actually get infected. And what I came to Chalmers specifically was to build a new robot scientist called Genesis. And the idea of Genesis is to industrialize experimentation. So it's going to have 10,000 little microchemostats, which will allow the robot scientists to plan and execute 10,000 experiments a day. And the application area is to try to understand eukaryotic cells. So humans are eukaryotes, so it's trying to understand human cells. And only an AI system could organize the work of doing 10,000 things every day. That's way beyond what any human being could do. So this is a little initial module of Genesis. The first part should come this month to Chalmers. And just to finish off, I want to say a little bit about the future. So there's an analogy between science and games, I would say, and that in chess and Go, these games, there's a continuum of ability from very simple players like myself up to the grandmasters. And computers follow that path. The first computers playing chess were very poor. Now my phone can easily beat the world champion at chess. And I argue that this is also true in science. There's a continuum of ability of science from the very simple types of science that Adam and Eve can do up to the type of science that you and I can do, up to the sort of grandmasters of science, the Newtons and Einsteins. And if you accept this analogy, then I would argue that it's very likely that computers will get better and better at science, have better hardware, better software, more and more data is going to be online for them to work on. And so they're getting better, and humans are not getting better at science. And about 10 years ago, the Physics Nobel Prize winner, Frank Wilczewicz, said that in 100 years' time, the best physicist will be a machine. Just before the pandemic hit us, we had a meeting in London on what's called the Nobel Turing Challenge, which is by the year 2050 to build a machine which can do Nobel Prize quality science autonomously. At the moment, it's a British, US, Japanese initiative, but we're looking to broaden this out to Europe and other parts of the world. I'd just like to thank all my collaborators. The DNA computing team is in the audience here, and my collaborators back around the world. And I'd like to thank you for your attention. Thanks very much. And I'll show this video of Eve in Chalmers. And I'm open to questions.
Yeah, sure.
See if this will...
I was waiting for a video.
It's not... Audio-visual people? Is it?
So,
here it's going, yes.
Yes, this is it.
Mm-hmm. We don't need to watch it. We can have some questions, or we'll have questions later.
No. I was just curious to see what was going to happen. You said that the robot scientist is getting smarter and smarter and works quicker and quicker. What about the human scientists that were working with those quick robots? Are you able to follow?
Well, at the moment, we're still much smarter than the machines. They're very good at certain things. They have sort of advantages over us in certain areas, but we're still a lot smarter than them at the moment. We know that we're doing science. The machine doesn't even know it's doing science. It's just solving problems in a particular area. So humans are still doing the big important things of deciding on strategy and what areas to work on.
So far you are a little bit smarter.
Yeah, we still have the edge.
Okay. I was just also curious, you mentioned something about that Eve is working on finding a medicine for against COVID-19.
Yes.
How far have you come in that research?
It's we found our first lead, so that's one of the first steps to do it. It's actually very shocking to me. Actually, at the start of the pandemic, I contacted everyone I knew in the big pharmaceutical industry. It's Big Pharma, it's called, yeah, and said, "What are you doing about finding a small molecule to treat COVID?" And as far as I know, very little was done. And AstraZeneca, they said they were not working on it, and I tried to. How is that possible? Surely it's not up to you to decide that anyway. Surely the Swedish government, the British government should tell you to do that, but that didn't happen. They worked on what's called antibody drugs, which means that rather than a small chemical which you could take to India or Africa or something, it's a very expensive approach to treating the disease. And why they did that, I'm not sure. Well, I can guess, but I don't want to say it out loud.
Mm-hmm.
Okay.
And then finally, just Eve. Does she always do like you want her to do?
No, last night she dropped something.
Oh.
Yes.
Sounds dangerous.
Yes, that happens.
Yeah. But you still have her under control.
Yes.
Okay. So looking forward to hear more from you in the panel. Thank you so much,
Okay,
Ross.
thank you very much.
Now I would like to turn to Stefan Jansson, who is a professor and joining us from Umeå. Where his research is about plants. And he will talk about the methods he uses to modify the genome of plants. And one of them is CRISPR, the genetic scissor, as I suppose you heard about last year in connection with the Nobel Prize. Most welcome, Stefan.
Thank you. You hear me okay?
Perfect.
Perfect. Okay, thank you. I'm immensely honored to be part of this event here, and I will, as I said, talk about this for a few minutes. I should start by saying that perhaps you heard this before because it's been clever men and women who have said that the 20th century, that was the century of physics. That brought us computers, that brought us a lot of energy and so on. But maybe the 21st century will be the century of biology. And if that will turn out to be true, then CRISPR-Cas will definitely be one of the tools that will make this possible. So I will talk about this, but in a little bit wider context, and I will now try to share my screen, see whether that will work. It will take a few seconds for my PowerPoint to start. So, but hopefully you will see it when I will see it. So, CRISPR is, as Helena said, a genetic scissor that can be used to reprogram organisms. But before going in a little more to the details what CRISPR can do, I need to start somewhere by talking about what is really a gene. I mean, of course, most of you have some knowledge about that. You remember something from your high school and so on. But really, just to remind you, I mean, we have long stretches of DNA in our cells. We do it, humans, animals, plants, bacteria, and so on. And on this string of DNA, there are bits and pieces that have a biological function, and that's a gene. What genes are doing, as Ross just said, that's just keep some information, because the gene is translated into a molecule called an mRNA that will make up a protein. And in almost all cases, it's the proteins that does the work. So you could have one gene here and you have another gene here, but it makes another protein. And the classical example that was used in genetics courses when you were in 9th grade at school, that was eye color. That brown eyes and blue eyes, that's a genetically determined trait. And really, so what everyone probably listening to this now, they have a gene, say it's this gene, that produces a protein that produces a blue pigment in the eye. That's why we have blue eyes, but people that have brown eyes, they have another gene somewhere, on another chromosome in fact, here I'm true, that will produce another protein that makes a brown pigment. And the brown pigment in this case is sort of stronger than the blue pigment. So the brown-eyed persons, they have both the blue and the brown pigment, but you will see this as brown eyes. So we who are blue-eyed, we really have a mutation in the gene coding for the brown pigment. That's why we are brown-eyed. So mutations, differences in these genes that may then function or not function, or function in different ways. That's what makes us different. What makes me different from a tomato and what makes me different from you. And a gene, it actually doesn't say. So if you would read the DNA, there would be a lot of A's and T's and C's and G's in a long row. It doesn't read make eyes blue. It actually reads TGA and so on for perhaps 10,000 letters. A long gene maybe 10, 20,000 letters and so on. And that is the code for making the protein that has this specific function. So I work with plants, and our main model plant is aspen, the normal Swedish aspen. They are, of course, growing in the forest, and this time of the year you can quite easily tell one individual apart from the other, because aspen, they can also make a lot of shoots, so there could be sort of 10 or 20 or 100 trees that really are the same individual, but they are different. And in the autumn, this particular one, it starts getting yellow leaves 2 weeks before this one. So this is one individual and that's one individual. So we've been looking at the genes and the genomes of hundreds of aspen trees. So from now we know very good data on that. If you take an average gene and look at this, and this individual and this individual, they will differ by about 1%. So for 10,000, if one gene is 10,000 bases, letters, then about 100 of those will be different if you compare this one to this one. Maybe this doesn't say too much to you if you're not a geneticist. But as a comparison, if we would compare mine and Max DNA, we would find that they will be about 0.1% different. That's the average difference between humans to humans. It will maybe, since we are from the same population, maybe it's a little bit less than 0.1%. If we compare me with someone, an aborigine or who knows, someone in Alaska or so, maybe it's a little bit more, but roughly 1.1%. So we are, the aspens, they are much more genetically diverse than humans are. We tend to think that we are so unique and so on, but that's just because we spend so much time and energy on trying to look different. We have different clothes, different haircut and so on. And also since nature has made our faces a little bit, or very much different. That's because we want to recognize each other, because we are social animals. The aspens, they don't care to recognize one from the other, but they have been developed to be much more genetically diverse. The reason is really that up to humans started to make railroads some 150 years ago, we can basically only mate with those that are within walking distance. So, so we humans are very much inbred compared to many other organisms. However, if you would compare a human and a chimpanzee, two different species, then we will see that the genes are about 1% different. So the two aspens that you would find if you out in the forest, they are as genetically as diverse as a human and a chimpanzee, which is quite amazing to start with, and it also means a lot of biological information that we can extract from this. If we can understand what makes, for instance, this gene, this aspen being yellow much before this one, this is our main research project at the moment, trying to understand the genetic basis of why one is getting yellow before the other. How do they know that autumn is coming? So we have a lot to do here, and we can use that later on for different interesting things. So Ross talked about cabbages, and here you have a lot of cabbages. So I'm using this picture to illustrate what we have been doing during our history as farmers. I mean, humans started to with agriculture about 10,000 years ago. And since then, we've been using plants to grow next to where we live and have developed different types of plants, new cultivars. And up to about 150 years ago, we did this without knowing anything. We didn't even have a word for genetics. We didn't have a word for breeding. But still, we have done amazingly a lot of things. This is the wild cabbage growing in the Mediterranean area. It looks like this. Been cultivated for thousands of years. Once upon a time, one farmer in a field happened to find an individual cabbage that had a mutation in a gene that was necessary to make branches. So instead of making branches with a lot of leaves on, the branches was only 0.5 centimeter long. So all the leaves came at the same place, making a little ball like this. So that particular farmer thought, this looks nice. He tasted probably this one, he or she, and thought it was good. And then he took seeds from this individual, and the seed and the new plants had similar structures. And today we eat the offspring of these ones as Brussels sprouts. Another farmer somewhere found an individual plant that had a mutation in a gene that was necessary to make a stem. Today we eat the offspring of that as Brussels sprouts. Cauliflower, this is actually the part of the plant that should become the inflorescence with all these flowers. Here it's swollen up more or less like a tumor. That tastes good, so that's why we are growing this one and buying it in the shops. All these ones they are the same plant species. That's why we can cross them with each other and generate a lot of new varieties every year. And that is why there comes new things in the grocery shops every year, new varieties of plant due to plant breeding. Exactly the same has happened with dogs. We've been having dogs, domesticated dogs, for thousands of years, and they have been diverging in different directions depending on how we have used them and so on. We have selected them and made now, so we have very many dog breeds. That was also done without knowing anything about genetics. It was just, it happened to be. So just selection by humans is an efficient way of change animals and plants. So, but all these things with cabbage and other plants and dogs, up to a couple of, not so long ago, we really had to sit and wait for the random mutations to happen in the genome that we later could use to breed for new varieties of plants and so on. So it was, we could live with the variation that was there, that nature has created, but we couldn't do much about it. But now, since about 7 years, we have a much better tool actually to order these mutations. And this is CRISPR-Cas. And we, in Umeå, we are immensely proud that Emmanuelle Charpentier was working at our university when she made this, the real discovery here. Of course, she moved to Germany before she got awarded the Nobel Prize, but okay, we can still take a little, claim a little fame out of that. But so, and this was already back in 2015, dubbed as the scientific breakthrough of the year. So what is this? How could we use CRISPR to change the DNA of organisms? Well, CRISPR is really a part of the defense of bacteria towards viruses. And this is what Emmanuel and many others were studying for decades, trying to find out how do bacteria defend themselves against viruses. And they found out a lot of things and elucidated this whole pathway. This you don't have to worry about now. The only thing which is important for genome editing that we are using is this little piece here. This is a protein and enzyme that cuts DNA. There are several different Cas proteins. And this protein can be programmed to cut DNA on a specific place in the genome among the billions of bases that are there by adding a little piece of RNA that is sitting here. It's bound to this Cas9, and then it finds it's matching one piece of the DNA, and then it makes a cut here. So that's how we make a targeted mutation, an edited mutation, which we can, again, if mutations were random, they will only happen once every 1 billion times where we want them. Now we can make them happen every time where we want them. So, and of course, that could potentially be used and has started to be used to correct genes in humans. Those of us that have a genetic disease basically have an error in some gene that makes it so it cannot make what it's supposed to do. That could be corrected. Here there is a genetic heart mutation that they injected these things into an embryo, got the CRISPR to edit this particular gene that was not as it should, and then got back to what it was supposed to do. So it can be used in human cells, we know that very well. Then we will probably discuss later on whether we should use it. But there are other interesting examples. There is a group in the US also that have taken on the task to, could we recreate the mammoth? So they use the fact that mammoths are relatively genetically similar to elephants. So of course there is not one gene that differing from an elephant and to a mammoth. There are hundreds, probably thousands of genes. But if you can find out which are the important genes, you could potentially go through them one by one and eventually start with an elephant embryo and make a mammoth. I must say, I don't know whether they will ever be able to do this, but this is this type of projects that half crazy scientists can start doing now. But the good news for people like me, which is doing basic science, is that making the change, that's the easy thing. So the hard thing is to find out what change to make. So like in this case, I said that we have a research project is trying to understand which genes that makes this one starting senescing 2 weeks before this one. We have been studying this for 20 years. We are still not there. So, and if we don't know which gene that should be changed in which direction, which T that should be changed to which C, for instance, then we can't address this with CRISPR. There are other cases where we know better. We have, for instance, studied also in aspen the genes that tell whether the aspen should set bud and stop growing earlier in the autumn, earlier or later. And here we've been able, by doing genetic screens, to identify, we know which gene, it actually has the name FT2, and we know that all aspens in northern Sweden, they have one variant of this, they have a G at one certain position, and the aspens in southern Sweden, they have a T at this certain position, and those aspens in the middle of Sweden, they have some have a T and some have a G. And that renders that these trees growing up here, they set bud under very different conditions. They need to set buds earlier in the autumn because winter comes much earlier here than it does down here. They have been a local adaptation. So all trees up here, only those that have this particular combination of genes, they can survive under conditions up in Umeå. So, and if we know this, then we can go in and change. So we can, in this case, we can destroy this particular gene so it doesn't do its function. And then we can shift one type of aspen to the other type of aspen. So, oh, what happened? So, but we need to know in advance what to do. So we have spent years and years on this, and once we know this, then it's a piece of cake just to do the change with the CRISPR. And that's the nice thing with CRISPR. CRISPR is not the first technique that could be used to change the DNA of an organism, but it's much, much easier and much more convenient. So before, it was only very few cases where scientists used this. Now we can do it almost everywhere. So, but the other limitation, of course, maybe needless to say, is genes don't affect everything. There is as a biologist, I think that we most agree that about half of our traits are governed by genes and the other half by environment, and most of them are in interaction. So for instance, if we would like to make better hockey players, there we would have a problem, because we still don't know which genes that makes one person better hockey player than the other, because there are hundreds of genes probably involved there. It doesn't help to just have bigger muscles or be quicker on skates and so. You need to have a lot of combinations. So we don't have genes, we don't know the genes for being good hockey player. But the other part of hockey, for instance, whether you support Sweden or Finland, that has nothing to do with genes at all. It just happened to on which side of the border you happen to be born. So that's clearly a trait that is encoded, not encoded in genes, but is dependent on environment. And those we can never address with CRISPR and whatever thing we are doing. So, but maybe I should stop here, or what do you say, Helena? Yes, we will come back later on to these questions.
We will come back later on. Of course, you will participate in the panel discussion. I just wanted to forward a couple of questions that we got on Menti to you, and they are almost the same, and they are similar at least, because they are asking, why have you chosen aspen and not another tree? Is it something that has to do with the conditions of an aspen, or why? Tell us.
Aspen is the best organism to work with for our purposes. And that's because it's most easy to propagate. Everyone had an aspen in your garden and tried to take it down. You know there will come 1 million of small aspens coming up.
Sure.
So this means that we can make hundreds of copies of the same genetic individual very easily. And that was also the first tree that we could genetically modify with the standard techniques. So that's why it's the best model system.
This about changing, affecting genes or working with modifying plants and so on has been a subject for the political discussion during years and years. Where is it now? I mean, it isn't that much debate about this nowadays. Tell us.
No, well, now everyone talks about COVID. No, but from my perspective, almost nothing has happened in Europe for the last 20 years. We had a legislation that basically prevents us from using our knowledge. I mean, we have been generating plants. We basic scientists have found out traits. Breeders have created plants that have basically whatever trait you would like for a plant. More nutritious, less affected by, and so on. But since it's been regarded as a GMO, so by law and by consumer preferences, these are not used anywhere in society.
But there are sorry.
Yeah, and this is really why CRISPR became so interesting, because the whole definition of being genetically modified is in most countries that you take a gene from one organism and put it into another. If you only take and change one letter of the gene. When this technique was developed, as I said, in 2014, then it was not obvious. Would that become a GMO or not? Because it really depends. If it would be a GMO, it cannot be used. If it would not be a GMO, it could be used by anyone everywhere. So, and now, maybe we'll come back to that later in the discussion, but the situation right now is that in Europe, this is regarded a GMO if we only change one letter using CRISPR. In the rest of the world, it's not. So it makes it almost, or it's definitely an impossible situation for those that would like to keep up with these restrictions, because something which is not a GMO elsewhere will suddenly become that if it will be carried into Europe. And the problem is that we can't tell them apart, because if we make this particular mutation by CRISPR or whether it's happened naturally, they are absolutely the same. So there is no way to know whether the particular plant is illegal or legal.
Interesting.
Which is no good news for the police and the custom.
I suppose so, yes. Very interesting. Thank you so much. And we come back to you, of course, in the panel discussion. Now I would like to welcome Elin Esbjörner. Elin is Associate Professor in Chalmers of Chemical Biology. This was difficult. Elin will tell us about how to develop new, more efficient, and more pharmaceuticals based on new technologies. Most welcome, Elin.
Thank you. So, my name is Elin, and I research essentially two things. I'm interested in what happens in the brain when you get Alzheimer's disease, that I will not talk about today. I'm also interested in how we could improve mRNA therapeutics, so a type of genetic drugs that I think you now all have heard of because of the success that these have had in the COVID vaccines. And the reason why I'm interested in them is because mRNA, I pressed the wrong button. mRNA therapeutics, they encode for proteins, and proteins are responsible for almost all the functions in our bodies. And when we get a disease, that is almost always because a protein goes wrong. And the mRNA and other RNA therapeutics, they can be used to correct this faulty protein. And a major task that we have to enable mRNA therapeutics is to actually get this drug into the cell, as is illustrated here in a drawing that my collaborator Fredrik Hökes made. So we study nanocarriers that can take mRNA therapeutics into the cell and enable their function. And before I tell a little bit about what we do, I just thought I'll tell you a bit about the COVID vaccines that I think most of you have probably had. And I'll tell you what you actually got when you got the COVID vaccine and what it did to you. So the COVID vaccines, they contained mRNA. And this mRNA was actually packaged into a lipid nanoparticle, a little fat droplet. The mRNA was injected into your muscle cells, where the ribosome translated it into the spike protein that sits on the surface of the coronavirus. The muscle cell would then transport the spike protein to its surface, where it could be recognized by the immune system, and you got, hopefully, antibodies to the spike protein, so that the next time your body encounters the virus, it has protection. So the mRNA therapeutic, it encoded for the spike protein, and it produced it in your muscle cells. And the beautiful thing with this technology is that, and this is just an more sort of fancy illustration of how this works, because the beautiful thing is that your body can produce any protein by reading the mRNA in the ribosome and producing a protein chain. So if we think about this, not the spike mRNA being a spike protein, but actually we could produce any mRNA and we can make any protein. And with that prospect, the possibility to treat disease, it becomes essentially limitless. And there are also other sort of sister technologies to mRNA therapy, because we can also, instead of delivering mRNA, we can deliver small, for example, siRNA, but these drugs can have many names, and they turn off protein expression. So we have a complementary system. mRNA, it turns on protein expression. Here an example of delivery to the liver. The mRNA goes into liver cells, gets translated by the ribosome, and it produces a protein that the liver needs to function. The other option, if we use an siRNA, we can also deliver it, in this case, to a liver cell. The siRNA that will go and find a particular mRNA and make sure that the cells degrade it. And in that sense, you will not get a protein. And this particular example is one of the first approved siRNA drugs against the disease called TTR amyloidosis, or in Swedish more known as Skellefteåsjukan. It works by reducing the amount of this TTR protein that goes on and forms fibrillar aggregates in our body and leads to all sorts of problems in many organs. Common to both these technologies is that we would never be able to get the RNA into the cell unless it was packaged. And the packages in both cases are quite often the lipid nanoparticles that we study. And these are actually quite easy to make. You take lipids in an ethanol solution, an mRNA in a water solution, and then you put them down this type of device that has a T-junction, and then you push, and it goes some twists and turns, so you get good mixing, and out you get the lipid nanoparticles. And if you look at them in an electron microscope, they look something like this. So they have a protective layer, and then they have the mRNA inside. And because of that, the mRNA is protected, and that's one of the main functions of lipid nanoparticles. They need to protect the RNA so it doesn't degrade in the biological fluid before it reaches your cells. Ideally, you would also want to target these lipid nanoparticles so you can get the drug to a specific organ or a cell. In the case of the COVID vaccine, that was solved quite easily because you injected it directly into the muscle. But for other diseases, maybe you would want to make an intravenous injection, and then the particle needs to travel through the bloodstream, gets to the correct organ and to the correct cell within that organ. And then these lipid nanoparticles, they also need to facilitate uptake into the cells. And this is a major challenge in RNA therapeutics. And at Chalmers, we actually do quite a lot of research in this area, and it's not just me. So we have a research center that's called Formula X for formulating RNA carriers inspired by nature's own way of delivering material that are called exosomes. And this is an SSF-funded initiative that started actually a few years before the COVID pandemic. And here are the people involved and the companies involved. We have also done some innovation within the company. So a company spin out by Fredrik Höök that's called Nanolize, and that makes instruments. And me, together with my colleague Marcus in chemistry, we are spinning out the technology to introduce more efficient labels into the mRNA so that it becomes easier to visualize when developing drugs. So what do we actually do to try to make better lipid nanoparticles? My group, we focus on live cell imaging, and I will soon show you a little video here. Because we believe that if you can see how a lipid nanoparticle is taken up and perform its function in the cell, then we can understand better the mechanism by how they do this. And if we understand the mechanism, we can design the better particle. So here are 3 videos with some different lipid nanoparticles. They differ in size, or they actually differ in the lipid composition of the outer layer. And what you will see in these videos are the lipid nanoparticles with the red fluorescent mRNA, and eventually they will encode for a fluorescent protein that you will see in green. And you will have to have a little bit of patience, because when we add this to cells, things doesn't happen immediately. It takes some time. But here you can see how the particles starts to get taken up, and afterwards the cells start to express the protein. And here you can see that these 2 particles, they are pretty efficient. You get a lot of the green signals, so a lot of protein in these cells. Whereas this particle that actually differs from this one only by 5% in the lipid composition, it doesn't work at all. Or actually, if you would have looked at this on a slightly better screen, you would have seen a faint green signal in the background. So by tweaking just lipid compositions a little bit, you can make a huge difference in how lipid nanoparticles function. Also, if we take this lipid nanoparticle that worked so well in the last image in the delivery to liver cells, but we instead added them to an immune cell, a dendritic cell, you can see that in this case, the cell gobbles up a lot of these particles. There's a lot of red signal here, but it's absolutely no GFP fluorescence. So the lipid nanoparticles are also very cell-specific. So if we want to target the immune system, we need a particular particle. If we want to target the liver, we probably need a different one. But right now, we don't understand how to design one or the other. And the difference between different cell types, it lies in this process of the cellular uptake of the lipid nanoparticles. They go in via a process that's called endocytosis, which is essentially, you could see it as the gut of a cell. So they first eat something, and then they transport it sort of downwards towards the intestine. And while they do that, they also degrade the particles. So somewhere along this path, the lipid nanoparticle, it needs to escape out in what is called the cell cytosol, where proteins can be produced. And in the video I showed you before, you can see the uptake in red, and you can see the protein translation in green. And these processes are quite easy to monitor. But in order to fully understand how LNPs function, how they deliver their mRNA, we also need to understand the process of when the mRNA escapes from this endosome or the gut system and gets out in the cytosol. We have developed a method to do so, which is based on a fluorescent protein that gets recruited to structures like this when they break and release RNA. So if these structures break, you will get red dots in an otherwise faint red cell. And here this is actually depicted in yellow instead. These are cells that have just been treated with a small drug, this chloroquine, that is actually used to treat malaria, but that has very powerful functions on the cellular level. And you can see that you get lots of dots in these cells, and every dot corresponds to a rupture or broken endosome where the content can escape. So with these tools, we can have one signal for the uptake, one for the escape, and one for the translation. And what we do in this Formula X consortium and in collaboration with AstraZeneca is that we screen a lot of lipid components. And at Astra, they can make them by robotic systems, and they can also image thousands of formulations at the same time with other robotic systems. And then we can screen to optimize performance. And I will exemplify this by 3 examples. So this is delivery over time, so the protein production over time. And we have here 3 different LNPs. It doesn't matter so much what they are, but what matters is that in each set there is a difference. So here is one that produces very little protein, and this one produces a lot of protein. And if we look in the top one, when we measured the uptake, we could actually see that there was a difference in the uptake levels, and there was a difference in the escape levels. And probably these two particles, or the three particles actually here, they were different because they are taken up differently by the cells. But then we can also have example that looks like this, that these two particles, three particles, they were taken up, two of them were taken up exactly the same, but they have different capacity to escape from the endosomes, and that's why we saw differences in the protein production. And then the third scenario is that we have particles that can be the same in uptake, they can have the same efficiency in this escape, but they still produce different amounts of proteins. And then they can have a problem in the translation when the mRNA has reached into the cytosol. And that could, for example, arise if the lipid nanoparticle holds on too tightly to the mRNA and doesn't release it once it gets into the cytosol. So by doing this type of screens, we can get biological insights into what LNPs function in what cellular system and what lipids are important to include in a good lipid nanoparticle. But we also like to understand in more detail what actually happens, because this doesn't tell us why a certain lipid is good. It just tells us that it is good at certain traits that are important in delivery. So in Formula X, I just showcase a couple of examples how we do to better understand why. And we, for example, combine the cellular studies with mimetic systems that we can study in vitro, so that we can model this endosome from which the mRNA has to escape and understand how the lipid nanoparticles interact with the endosomes. As I mentioned briefly before, we developed new labels so that we can better study the mRNA, because the labels I've shown you today, they are based on a quite large fluorophore that you hang on to the mRNA and that disturbs it. But we can also put on much smaller ones that we call stealth labels. We are also very interested in what happens with the lipid nanoparticle when it encounters biological fluid. For example, when you inject it into the blood and it meets plasma proteins. Because it turns out that these plasma proteins, they will go on and bind to the surface of the lipid nanoparticle and form what is called a protein corona. And this protein corona is absolutely decisive for uptake. And some of the proteins that form this corona are, for example, lipoproteins that we know from if you have high or low cholesterol, for example. That type of proteins are very important for the delivery. And that also implies that maybe your body composition, whether you're obese or not, whether you have some other illness, maybe fever or not, can affect how well you respond to lipid nanoparticle. So therefore, we're also very interested to understand these type of proteins that attach and what role they have in the delivery, to be able to make more personalized treatments and make sure that the patient that gets an mRNA therapeutic actually responds to it. And then we also study nature's own type of delivery vehicles, and they're called exosomes. We try to engineer them to contain RNA drugs, and we also just try to learn how are they composed, because these are incredibly efficient messengers between cells in nature, and they are almost always non-toxic. And if we could learn from them and bring that design to the LNPs, we believe we could make much better mRNA therapeutics in the future. And with that, I'd like to thank, just sum up a little bit, to tell what our strategy is. So we want to generate knowledge of how LNPs function and mRNA is delivered, because we believe that if you understand, you can rationally design something, and then we could provide effective delivery solutions, and mRNA therapeutics could have a broader clinical translation beyond the mRNA vaccines, which we think are actually just the beginning for this therapeutic area. And of course, you never do research on your own, so these are some people I want to thank, and the people involved in particular are people that are involved in the Formula X project. And I'd also like to thank you for your attention.
Thank you so much. And if I understand you right now, what you're doing will possibly lead to more customized, tailor-made medicines in the future. Does it mean also that the future medicines will be able to design medicines that are taking into account the age, the weight, the habits or whatever from different persons?
I think potentially one could do so in the future. I think what is nearer at hand is, say that you have a cancer, a particular type of tumor, then we can figure out what tumor do you have, and we can design the mRNA so it fits exactly for you. I think that is the first step in personalized medicine. But also, as I said, for the delivery of these particles, we know that the efficiency of the medicine will depend on your body status.
Mm-hmm.
And there we might actually need to adapt, because otherwise there might be populations that cannot respond to the medicine at all because they have the wrong set of proteins in their body.
But this could also mean, if I understand you right, that in the future it will be possible to reduce the side effects, because I mean a lot of pharmaceuticals have a lot of sort of side effects, and I suppose this will lead to less side effects in the future.
Yes, and no.
It's always like that with researchers, you know. They never say yes or no.
mRNA therapeutics, they can also have side effects. They can have quite strong immune effects. Maybe you got a fever when you got the COVID vaccine. And maybe that's okay, because you're only going to get the COVID vaccine twice, right? But if you're going to have an mRNA therapeutic for the rest of your life, for lifelong treatment, say you need an injection per week, you don't want to get the fever every time. And that is why it's so important to make them more efficient.
Talking about the vaccine, I got a Menti question as well, as somebody asking, what is happening if you get an injection with the vaccine, not in the muscle, but in the blood? Somebody who's worried, I suppose. If they get by mistake, if you're given the vaccine in the blood instead, what will happen then?
I don't think so much would happen.
Okay, so whoever wrote that question could be calm then. Thank you so much, Erin.
Yep.
So now we are going to continue the discussions that Max gave us such a lot of ideas and things to talk about. And meanwhile, I have also got several questions on Menti. And Max, the first question is to you. Do you believe human cognition is insufficient of full comprehension of reality? And do you think that is an argument for strong AI? What do you say about that?
So they're kind of two separate questions. Do I believe that it's impossible for us humans to ever fully understand the nature of reality or not? Mm-hmm. I tried to be very humble about this, and I'll say I just don't know. But what's really remarkable is how much more we understand today than we used to, if you think about it. And I think there's no better way to fail to understand things better than to convince yourself that it's impossible. So I think it's a healthy aspirational assumption that we can understand a lot better, so we try harder. Then the question came in about, is this an argument for stronger artificial intelligence? Well, I think everything we love about our civilization is a product of intelligence, right? So if we can amplify our human intelligence with artificial intelligence, of course we can use it to solve all sorts of great problems, cure all sorts of diseases, as we've heard about, and help eliminate poverty and so many other things. But we can also use it to drive humanity extinct by doing something stupid. So I think we can never separate the question of whether we should be for more technology or not from the question of what we're going to do with it. I love technology. Chalmers Tekniska Högskola has the word technology in its name, and Massachusetts Institute of Technology also. But I think it's irresponsible for us, those of us who keep making technology better, to not also think very hard about how it's used. and make sure that we actually go out and advocate to those who make the decisions for it being used beneficially, not harmfully.
And let's discuss that. How can we then use the technology, develop technology, but still have some sort of control? What do you say, Stefan? What is needed?
Yeah, I don't know. I'm just so happy that I'm a scientist and not a politician, because at the end, all these things have to be decided. I mean, there are so many conflicts of interest and so on that has to be balanced against each other. And it's easy for us scientists to say, this is what we can do, this is what cannot be done, and so. But it is very hard. I'm also very humble about very hard for us to say what needs to be done and what should be allowed and so on. So I don't have a good answer for that.
As a scientist, do you wish that you had some guidelines, some sort of frames telling you what you should do or not do? What do you say here, Ross?
Well, Stefan talked about the policy in the EU on GMO plants, which, as he pointed out, is not very wise. I think and it's not one of the. I'm completely against Brexit, but one of the benefits to the UK of Brexit is that they're going to get rid of these rules. So having rules is good as long as they're sensible rules. And I think that was European politicians made the wrong decision at that point.
What do you say, Eli? Do you feel that there are limits, or do you wish that there were more guidelines?
I don't see that there are limits to science and our ability to discover things. But legislation, if it gets too old, I mean, the legislation has to adapt to what we know and how safe we know that various systems are, if we talk, for example, about GMO. And in Europe, it is too restrictive, I think, today.
But is it so that the decision makers are running after, or are they aware of what you are? I mean, do they have the knowledge, to put it that way? What do you say, Max?
I think that's exactly the problem. The problem is not with the scientists. The problem is that the politicians don't listen much to the scientists. They don't listen much to you. There are much more corporate lobbyists in Brussels than there are scientists. And just go look at Sveriges Riksdag. How many people in there actually have any kind of education in science, right? The answer, I've done the math. It's equally sad to the answer you get if you look at the American Congress here. So when you take an issue like, should Sweden actively in the Geneva negotiations right now about these slaughter bots, right, that hunted down humans in Libya and the machine decided who to kill, should they be banned or not? Right now, the Swedish politicians seem to be listening more to the weapons lobby than to us scientists, because the scientists in AI are horrified and say, we don't want this to be done in our name, this should be banned. But the Swedish government, I'm incredibly embarrassed by how spineless the Swedish representative is right now in Geneva. We were just there last week. And they're not listening to us, And so maybe things like this symposium is exactly what we need more of, where there's a forum for scientists to talk about these things and get politicians to listen to what you say, not just the lobbyists who try to sweep concerns under the carpet.
But another problem nowadays is also that people in general don't listen to you as scientists either. I mean, you have the anti-vaccine groups, for instance. You have a group of people who don't believe in the scientists from IPCC when it comes to the climate.
Yeah.
How do you look upon that as scientists? I mean, what can you do to make people understand that what you say are important?
I think the reason why there is so much anti-science sentiment is exactly the media polarization that I talked about. The reason that people don't listen, that a lot of people believe that climate change is a hoax and don't want to listen to scientists, is because there was a very successful lobbying campaign run by large companies trying to discredit scientists. And guess what? It worked, right? 40 years ago, we saw exactly the tobacco industry do exactly the same thing. trying to discredit the scientists who said that smoking caused lung cancer. And then we had companies selling asbestos, trying to discredit the scientists saying asbestos causes cancer. And then they try. So if we let companies again and again demonize whatever category of scientists are inconvenient at the time, we shouldn't be surprised that eventually it's your turn, whatever your field is, that they won't listen to. All right. Now it's vaccines, for example. We can't just be so selfish and say we're only going to defend science when it's our science, when it's say vaccine science. We have to always defend it, and we can't tolerate a media system which is so corrupt that we let it be dominated. Why should Sweden, a small country that used to have a great respect for scientists, let its social media ecosystem be entirely dominated by American companies that are making Swedish people hate each other, so that this American company can make more money? Why is that in Sweden's interest? I have never heard any Swedish politician explain that to me, and I think many other countries would never even tolerate it. I can tell you, America would never tolerate that a Swedish company dominated the American media system and made Americans hate each other. They would just adopt the law saying, this is our country. We're not going to let other come in and make us hate each other. But so that's another thing Sweden can do, I think, just take more charge of its own intellectual
It's something.
Russ wants to...
Yes, I think, I saw some recent numbers of professions, how they're respected. And scientists are at the top there. Recently, the doctors have overtaken them because of COVID, I think. Somehow doctors have become more important. But they're still the most respected professions. At the bottom, they have lawyers, estate agents, politicians. But we are a respected profession. Of course, that doesn't mean that the public listen to everything we say, unfortunately. So it's not that the scientists are disrespected, it's somehow the message about certain cases,
It's a problem, definitely. But what I would like, you pointed out something before, and Stefan did as well, that there is a sort of a ban to research on the fields that you're working in. What consequences will that have?
It's not a ban on the research.
It's the ban of using the research in commercial applications.
Okay, so you can still make a lot of research, but it never reaches the audience or the people or whatever. Is that the case?
Yes. And that's partly political, but partly also it's the law. So the European Supreme Court decided how to interpret a particular law in one way, which
and
that was a big surprise,
are they misinformed?
I think they could have interpreted it differently.
But then I must ask you, as researchers and all of you, then what kind of responsibility do you have to make politicians and ordinary people and everybody to understand better, to not be against vaccine and to not be against important research and so on? What can you do yourself to inform us stupid people a little bit better?
Yes, we have to do what we're doing right now, participating in these events and so on. But in general, one shouldn't be too negative about this. We easily point out the things where science communication have gone wrong. But by large, I must say that community follows scientific advices and breakthroughs in most cases. There are certain cases where things go wrong. And that is, I think it a lot have to do what Max was showing there, this polarization. It's obvious in the US where you have two parties, either you are blue or you are red. And it is really hardwired in our brains, since we are social animals, that we would like to get together with our group, with our tribe or so. So this is why, as soon as things get too complicated, we tend to just take the opinions of the group that we want to belong to. And then you buy the whole concept. You get all the views and all the ideas from that crowd. And this is what really is so obvious in the US, where you have this, you can't be half. But we also do that in most other cases. And sometimes it goes wrong. As I said, the vaccine is one obvious thing, but I would still say that in most medical science, I mean, this is not an issue at all. I mean, most medicines are going out and people are taking them happily. So it's we shouldn't overemphasize the problems.
I want to just compliment Elin Esbjörner there. You answered this one question with yes and no, right? And our esteemed host was teasing you about it. But that's exactly what scientists do, which is the opposite of what you said the groupthink is, Stefan, right? That we are willing to be humble and admit that things are actually complicated and there are nuances.
Yeah, and I suppose that's the problem where you usually want to know what is right and what is wrong, and there are always both the pros and cons. Isn't that so, Elin?
Absolutely, and whereas we now discussed, for example, the GMO legislation in the EU, and we think it's too strict, we also know that countries that do not have any legislation at all, they do question genetic experiment on humans, for example. And that's also not something that we want to see. We want to make sure that before we do that, we should only change genetics in a person if it has a real purpose, and we should only do it if we know it's safe. So a certain amount of legislation we need to have until we know what is safe and what is not safe.
Mm.
What we not discussed so far, that's of course in the whole CRISPR field. I mean, changing plants is ethically totally irrelevant because no one has ever questioned whether it's okay that the carrots that we eat are orange instead of white as there are in the wild. So, I mean, no one cares about changing plants. And people care a little bit about changing dogs. Everyone likes different dog breeds unless they get to genetic diseases. So there we think we have the moral right to change the layout of the dogs if they don't suffer from it. But when it comes to humans, I mean, and this is something where it's, I mean, do we have the right? Do we have the moral right to change something? Because we all know you can't distinguish between curing a disease or making something much better in one specific direction. There is no black or white there. It's just a gray zone, which is ethically very complicated to know how to deal with. And that has nothing to do with CRISPR and so on. That has to do that we don't want to do breeding for humans.
I have to give you a compliment. I mean, both you, Stefan, and you, Elin. I mean, your fields in the life sciences have done a much better job at really thinking about where you want to draw the line between what's okay and what's not okay than artificial intelligence research has, where still basically everything is legal. You know, right now it's there's a Turkish company that sells these little slaughter bots that anyone can buy, and then they can program into them who they should kill and who's not, and who decides that? It's not the United Nations, it's the owner of the thing. It's legal. Should it be legal? Most AI researchers don't think so, but the Swedish government is opposing a ban. And should it be legal for a company, a large social media company, to just make money off of advertising that even though they know it just makes people hate each other more? We've had dramatic testimony by this brave whistleblower woman in America's Congress just last week. Some of you may have seen, right? Right now, it's completely legal. So you are our role model, actually. At least you have it. You have some rules, and then you argue about, well, maybe we have a little bit too much regulation, maybe we have a little bit too little. But at least you have the conversation. And in artificial intelligence, it's basically a vacuum still.
And you were talking about the great financial interest from Google and Facebook and so on to manipulate our minds. But it's also a lot of financial interest, of course, in all sorts of research. And one question here is from Menti is how to balance profits with ethics. It's a huge question, but what do you think about that? Who should, well, if I form it like this, who should own the research at the end? The universities or the companies?
Well, the universities normally sell it to the companies. That's how it works, you know. But I was once approached by a large tech company they would have paid me a lot of money, and it was an interesting application they wanted me to do. It was to listen to people while they were sleeping to do early diagnosis of disease. But which I could see that it could actually help diagnose a lot of disease, because your breathing patterns almost certainly indicate sort of starting of different diseases. But it's also clearly ethically a bit suspect to listen to people in bed. I wouldn't want me. Really, I don't want people listening in to me in my bed. But I could see it could really benefit people potentially. So it's hard to know always. What's the correct ethical thing to do there.
But obviously, when it comes to social media, it has gone. The financial interest has gone maybe too far and over the limits. But of course, we need companies to put the research into the market as well. I would also like to ask you to come into a little bit what we learned about Facebook and Google and so on. How can we then transform that into other areas? What can we learn from that so it won't happen in other areas?
I think the lesson is very clear. It's exactly what they taught me in Handelshögskolan back on Sveavägen once upon a time, that the free market is a wonderful way to stimulate entrepreneurship and make things happen and make them happen efficiently. If you have clear rules and regulations for what these come, so that the profit motive of the company will align with what's actually good for society, right? You don't allow a grocery store to sell poisonous food, for example, and it works out just fine, and as long as you have the rules and regulations. The problems we're seeing with Facebook and Google, and that we also saw with big tobacco earlier and so on, was that when the companies became too powerful, they started to be so good at lobbying that they could actually change the rules. They captured the regulators that were supposed to regulate them, and then the whole idea of why the free market was supposed to be good, right, stopped being valid. So I think, to me, the solution is the democratically elected institutions have to take back control and lay down, create a fair playing field again, where no company is powerful enough that they can, for example, dominate the media ecosystem and convince everybody that this company should not be regulated, right? So you instead align the incentives of the companies when they seek profits with what's actually good for society.
Let's ask the audience what you think. Let's go to your Menti tool, and we will have a question. Do you think that science and regulators together, or each one of them, will be able to handle the risks that occur with new technology? A very wide question, but whatever. When you start answering that question, I would like to put the question to you here. So what do you say, Elin? Do you think that science and regulators will be able to handle the risks that occur with new technology? Yes or no, or maybe? Or develop the answer.
I tend to be quite positive, so I think yes. But we might need some iterations.
What do you have? So you say yes, and why do you say yes?
Because I believe society finds way to deal with, let's say it's a disturbance and an imbalance in the system, and that we will find a way to deal with it.
Okay. And Stefan, what do you say?
Yeah, I guess I align with Max here that, I mean, big companies, they have their own agendas and their job is to earn money. And at one point, they get too big so that they influence politics. And we all understand that's a problem, but we don't know how to get around it and get out of it. So, I think, I mean, we've been doing pretty fine so far, and don't think we are going in the worst direction now than we've been doing before. But it's not easy.
No, or was it a yes or a no or a maybe?
Maybe.
Thank you so much, Russ. What do you say?
I think maybe. But I think we face so many, as I said in my talk, I think the world's facing so many challenges that we need better technology to deal with them. And they may come with some risks, but we have no real choice but to go down that route, I think.
So, Max, what do you say?
I think the best way to predict the future, which I will humbly admit I cannot do, is to look at the past. And then we see a very different answer depending on what field of science we look at. If you look at physics, we see we've done a great job in making sure people don't die in car crashes by putting in seat belts and airbags and many other things, and we have very safe airplane travel. Our skeleton in the closet is nuclear weapons. We right now have 12,000 hydrogen bombs, and Stockholm and Göteborg were almost destroyed in hydrogen bomb explosions, at least multiple times. When one time the only reason they weren't destroyed was because one particular officer on a submarine overruled the other Russian who wanted to launch. This was a misunderstanding, but we shouldn't have to play Russian roulette like that. So I think in physics, not doing so great because of the nuclear issue. Chemistry has been doing progressively better with banning a lot of toxins, especially in the European Union. The black sheep, the skeleton in the chemistry's closet is, of course, climate change. It did not happen because people didn't listen to scientists. It happened because, again, there were some companies, fossil fuel companies, who got so big that they were able to lobby and discredit people who worried about climate change. I think biology, as I already said, the life sciences is the one that's done the best so far. So all the rest of us should look to your successes and learn from them. I think artificial intelligence is the one that's done the worst so far, has failed to really make any meaningful regulation at all. The European Union is right now discussing GDPR for AI, so it's going to be very interesting to see if that turns into something useful. Unfortunately, the latest thing that happened was the latest version has been severely watered down by lobbying, so it's pretty meaningless. And I think artificial intelligence will ultimately become the most powerful of all these technologies, which is why it's so important that we get that one right.
So was that a yes or no or maybe?
The most interesting question for me is not to guess whether it's going to be great or bad, but to ask what can we do to increase the chances of it becoming good. And what I'm saying is we should look to life sciences and learn from it and do the same thing in artificial intelligence and physics and chemistry.
Thank you. Now we will look at the results of the Menti question. Yes, sure. Now Lena wants to join us here.
Can I use this? Today I read in Max is you still
If you
around?
come here, Lena, you can see.
Today I read in Dagens Industri lots of interesting things going on in the Chinese country about how the Chinese government is taking charge on control, not over the companies. We hear about Alibaba and all those things, but this is on all sorts of levels. Platforms on every level, every digitalization and everything which deals with commercial platforms. It could be security platforms, it could be media platforms. They're taking charge. And what you are saying, Max, is that we need countries to take on board the the responsibility for technology. The Chinese do. And they have very clever government. If you see the ministers there, they're highly educated, all of them. So is that what we should copy? I know you will say no. but there's something which I think is very, very interesting what the Chinese are doing. They are taking control. And we don't know which part of the world will be nicest to live in 50 years' time. So please give some words on this.
Was that for all of us? Or from...
No, it was for you.
Okay.
Congratulations.
First of all, it's so trevligt att ses igen. I think last time we met was in a taxi ride in Stockholm. To me, if you look at other countries, how they do things, the most interesting question, again, is not to ask, well, do we want to be exactly like that or exactly the opposite, but rather to look more nuanced and see what are the things that are actually, what are the good ideas we can take, and what are the things that we absolutely do not want copied. And I think you're highlighting something important there, which is the Chinese government actually has a lot of engineers and technical people in it. In fact, most of the politicians today still in China have a technical education, like many of you in the audience, which is very different from the way it is in the West. I think it would be really great in the West if we could just get more people with technical knowledge into the government. Because it's one thing to get technical people who they can ask, but it really helps also if the political leaders themselves can really understand the nuances of what you scientists tell them. I don't know how to accomplish it in the West, but it would be really delightful if we could encourage your ambitious students maybe to go into politics, some of them, or at least serve the Swedish government from time to time and let their voices being heard.
That would be great, I think. It would mean a lot. Now, well, let's have a look at what the Menti result says. Will the science and regulator be able to handle the risks that occur with new technology? Partly, well, that is, yeah. That is probably the way it is. We will see. Let's wait and see. Maybe with more scientists in the government, it will be even better. I would like to thank you so much, Max. I would like to thank Elin, Stefan, and Ross for participating this afternoon. Give them a big hand of applause. And then, to end this seminar, I will leave the floor to the chair of Chalmers today, Torbjörn Holmström.
Wow, applause. Thank you very much. Fantastic seminar, by the way, then, and thank you, Lena, for inviting all of us then to this fantastic seminar. When I was thinking about what to say, more than that you have done a fantastic thing for Chalmers, I was looking at an interview that you did 2017, which was very interesting. The headline was like this then: physics made my way into business. And then one of Sweden's most powerful board professionals. And working with you then during, I think it was 6-7 years then, I can understand that why that headline was there. Because you were extremely good as a chairman of the Chalmers University of Technology. And I've seen you, and I'm really, really honored then to be the one then that succeeded you then, and I have the honor then to run or to be the chairman. But Stefan talked about your history, everything that you have done. But when reading that article or that interview, everything you have done was that because it was fun. It was so fun to do. You jumped on that and that. It was fun to do that and that. And I went to European Union and so on and so on. So reading that, I thought that probably you were much ahead of Jeff Bezos then, talking about work hard, have fun, and make history. And you, by the way, you made history. In that article or in that interview, also they said that 1975, I was into lithium-ion batteries. And I understood that would be something for decarbonizing that mobility. And I phoned someone at Volvo and asked them, would you like to jump on that train? And they said no. I see Hans-Olof down there. I don't know if it was you, Hans-Olof, or someone else then at Volvo, or at the Volvo Group. It would be interesting then to hear who it was, but we could take that later on. So Volvo did a big mistake then, not then supporting you at that time, but for sure that was a groundbreaking thing then. So thank you very much for that. So the essence of your success, I think, is that you have energy, you are funny to work with, and you made history then, at least in my mind then. So I will not be so much longer. We are extremely grateful for what you have done then for Chalmers. and your contribution then to the successful university that we have today. And the rest of us will try then to take on the hat that you, or the shoes that you left, and try to walk then into the future. So thank you very much, Lena. Let's see if we have something. Could you come up? Oops, sorry.
Thank you, thank you. Thank you for the flowers, and thank you for the seminar. I think it was great fun. I couldn't have a better present than a seminar looking into the future. Problems, possibilities. So thank you all the speakers, and thank you, moderator. Helena, where are you? Somewhere. This is my old friend. Very nice that you moderated it all. And thank you, Mr. President. Thank you, Chairman. Without you, this hadn't come into place. And thank you for the years on the board. It was fantastic. We were part of recruiting Stefan, the success president, we can surely say. And we also made a lot of other things to support Chalmers in various ways when it comes to education and research. And before that, I worked a couple of years in the board of the foundation, which was new to me to learn about financing and how to see the legal issues at Chalmers, processing, how this is all set about. So that was interesting years. But all in all, I wouldn't be here if I didn't spend my 25 years at the physics department. Some of you are here, and I'm very, very grateful to those 25 years. So fantastic years. Tor Claesson, thank you. Björn Nilsson, thank you. Lars Brink, thank you. I would never have applied for a professorship if it wasn't for you. I remember when I came from ISIS, England, late at night, and you said, there is a chair in Uppsala, you should apply for it, do it tonight. And I did, and got it. So thank you very much, Todd. And also I want to thank my own research group, all the PhD students, and now you are professors, most of you, and you are out in industry. That was really great and fun, may I say, fun time, because it was fun time. So thank you all. Thank you for today. Thank you for the past. And Chalmers is in my heart.